Okay. This is the first question in 2006 paper. Balanda. Last day we discussed up to 2005. Now we can continue from 2006. First it asks, check your answers. Which one of the following is not a biological polymer? So remember, recall what are the four major groups of organic compounds: carbohydrates, lipids, proteins, and nucleic acids. Among these four major groups of organic compounds, polysaccharides, proteins, and nucleic acids are polymers. So therefore, RNA should be a polymer. Polysaccharides, proteins, and nucleic acids should be polymer. DNA is a polymer. Protein is also polymer. Cellulose is a polysaccharide, polymer. Starch is also polysaccharide, therefore it is also polymer. So answer should be ATP. ATP is a single nucleotide, so it is not a polymer. ATP is a single nucleotide, so it is not a polymer. Answer is ATP. Then second question. Which one of the following represents the monosaccharide composition of sucrose? Sucrose is formed by joining glucose molecule to fructose molecule. So answer should be 3. It's very easy. Glucose join to fructose and form sucrose. Answer should be 3. Those are easy. Then next, here there's a structured essay question. It says the above diagram, actually the diagram below, is a representation of a part of DNA molecule. Here you can see the 2 polynucleotide chains, and these dotted lines indicate the hydrogen bonds. The question asks to name the components labeled as A, B, C, D, X, and Y. Here simple A, B, C, D, X, and Y. Here you can see these adenine, guanine, cytosine, and thymine are nitrogenous bases. So this represents one polynucleotide chain. This is one polynucleotide chain. Clear? The other is the opposite polynucleotide chain. You know, complementary base pairing occur between 2 polynucleotide chains. Complementary base pairing occur between 2 polynucleotide chains. During this complementary base pairing, adenine, adenine binds to thymine. Adenine binds to thymine by 2 hydrogen bonds. Likewise, guanine binds to cytosine. Guanine binds to cytosine by 3 hydrogen bonds. In between guanine and cytosine, 3 hydrogen bonds should be formed. Another hydrogen bond should be there. In between guanine and cytosine, 3 hydrogen bonds should be formed. Therefore, I think you can easily understand this. Here, these dotted lines represent hydrogen bonds. These are the hydrogen bonds that form between complementary bases. These are the hydrogen bonds between complementary nitrogenous bases. Then it's very easy. If this is the adenine nitrogenous base, if this is adenine nitrogenous base, then simple A should be thymine. Simple A should be thymine. Mea thymine window. Simple A, thymine. Between adenine and thymine, 2 hydrogen bonds are formed. Here, 2 hydrogen. In other words, think like this. Here, these dotted lines represent hydrogen bonds. Here, you can see in front of adenine there are 2 hydrogen bonds. So at the point where 2 hydrogen bonds are present, at one side it should have adenine, and the other side it should have thymine. Therefore, complementary base to adenine should be thymine here. Similarly, here look at this. 3 hydrogen bonds are present in front of this guanine. Therefore, simple B should be cytosine. Simple B should be cytosine. Again, here capital C represents cytosine. Therefore, simple C base should be guanine. This is guanine. You can see here, thymine and cytosine are pyrimidines. Therefore, they have only single carbon ring. But guanine is a purine. You can see it has 2 carbon rings here. Carbon rings දෙකක්. Then this simple D should be adenine. Adenine, it is also purine with 2 carbon rings. X and Y, what are X and Y? X is the phosphate group. X is the phosphate group. Y represent deoxyribose sugar. Here, since this is DNA, don't just write Y as a pentose sugar, because specifically this is DNA. In DNA, specifically write the type of pentose as deoxyribose. This is deoxyribose sugar group. That is Y. Y deoxyribose sugar. Don't just write it as pentose, because this is specifically DNA molecule. In DNA molecule, specifically write the type of pentose sugar, deoxyribose sugar. Clear? Those are A, B, C, D, X, and Y. Okay, next it asks, write 3 features of DNA which are necessary for its function as a hereditary material of organisms. Last day also we provided the answer for this. As one feature we can say, DNA is able to, ability to store genetic information. Ability to store genetic information. One feature is ability to store genetic information. Second, ability to replicate, or you can say accurate replication of DNA. Ability to replicate accurately. Ability to replicate accurately, or accurate replication of DNA. Accurate replication of DNA. Then next, able to undergo mutations. Able to undergo mutations. Ability to undergo mutations. Ability to undergo mutations. Okay. So we didn't discuss those under chemical basis of life, because we have to discuss those under molecular biology in future. ඒවා ඉස්සරහදී කතා කරමු. දැනට ඔය past paper question එකේ හින්දා ඔය අර ඒ questions උත්තර include වෙලා තියෙනවා. ඒකින්ද අපි ඔය නිකන් කියාගෙන යන්නම්. Roman 3. මේක අහනවා name 2 laboratory techniques which are used to isolate DNA from cell extracts. This is also a question from unit 7 molecular biology. Therefore I will just give the answer. There's a technique, one technique called agarose gel electrophoresis. Agarose gel electrophoresis. Then the other one is, there's another technique called density gradient centrifugation. So actually this density gradient centrifugation is not mentioned in our syllabus now, but it is also a technique that we use in previous syllabus. Here we discuss mainly agarose gel electrophoresis. Agarose gel electrophoresis. We can discuss agarose gel electrophoresis in future under molecular biology. Okay. Next. What happens to DNA when it is heated to 90 degrees of Celsius? ඒක නිකන් ඇඩිෂනලි මම කියලා දෙන්නම්. ඔය ඔක්කොම හරි ඉස්සරහදී අපිට ඉගෙන ගන්න පුළුවන් molecular biology questions වලදී. මෙන්න මෙහෙම එකක්. Then when the temperature increases above 90 degrees of Celsius, or in basic media, or in strong basic media, DNA denature වෙනවා. Then we discuss only protein denaturation. Likewise, DNA can also denature, but the pattern is not same. Protein denaturation and DNA denaturation are different. During denaturation of DNA, what happens? Let's say this is the double-stranded DNA molecule. Imagine this is the double-stranded DNA molecule. I didn't represent this with a helical structure. So imagine these are the 2 strands of the DNA, double-stranded DNA molecule. And imagine these are the hydrogen bonds. Hydrogen bonds. During denaturation of DNA, what happens? During denaturation, what happens? Actually, denaturation occurs at temperatures above 90 degrees of Celsius. At temperatures equal or above 90 degrees of Celsius, or in basic medium. In basic medium, DNA can denature. During denaturation, what happens? These hydrogen bonds between 2 polynucleotide chains will be broken down. These hydrogen bonds between these 2 strands will be broken down. then two strands will be, or two complementary strands will be separated. Separated. Hydrogen bonds will be broken down. Then two strands of DNA will be separated. These are the two separated strands. So we can say this is single-stranded DNA. That means double-stranded DNA becomes single-stranded. That means two strands are separated. That's what happened. Clear? There are, as the answer we can say, hydrogen bonds between complementary strands break down. Hydrogen bonds between complementary strands are broken down. Hydrogen bonds between complementary strands are broken down. And two strands of DNA will be separated. Two strands of DNA will be separated. Two strands of DNA will be separated. That's what happened. Two strands of DNA will be separated. Two strands of DNA will be separated. Clear? Okay, that's what happened at 90 degrees of Celsius. But this DNA can renature. May renature වෙන්න පුළුවන්. ඒ කියන්නේ මේ opposite reverse reaction can also occur. මේක වෙන්න පුළුවන්. DNA can renature. Not like proteins. Then protein denaturation is irreversible. Protein denaturation is irreversible. If protein denature, it can't renature, but DNA can renature. That means when the temperature decreases up to normal temperature, up to favorable value, or if we neutralize the medium, these two strands again reform the double helical structure. That means when temperature decreases up to normal temperature, or when medium is neutralized, hydrogen bonds will be reformed in between these two strands. Hydrogen bonds will be reformed and it can renature. So DNA denaturation is not irreversible, it is reversible. Take a reversible. Okay. Those are actually in molecular biology in our syllabus now. අපි ඉස්සරහට ඒවා discuss කරමු. දැන් මෙතන ඉඳන් බලන්න part B1. Name two sites outside the nucleus. බලන්න name two sites outside the nucleus of an eukaryotic cell where RNA is found. In the nucleus, you know, RNA is synthesized. Outside the nucleus, one location where RNA present is matrix of mitochondria. Mitochondrial matrix has RNA. You know, mitochondrial matrix has DNA as well. So DNA molecule of mitochondrial matrix produce RNA. So as one location we can say mitochondrial matrix. Another location is stroma of chloroplast. Stroma of chloroplast. That means stroma of chloroplast also has a DNA molecule. Stroma of chloroplast also has a DNA molecule. That DNA produce RNA. In addition to that, you can write ribosomes. Ribosomes ලියන්න පුළුවන්. Ribosomes have rRNA. You know, ribosomes have rRNA. In addition, ඕන අපිට හිතමු cytosol ලියන්න පුළුවන්. In cytosol, then tRNA after synthesis, tRNA comes to cytosol. Mean tRNA comes to cytosol, which transport amino acids found inside the cytosol to ribosome. So in these locations, RNA molecules are present. Okay. Then Roman 2, name the site in the eukaryotic cell where RNA is synthesized. Name site where RNA is synthesized is nucleus. Majority of the RNA is produced by nucleus because most of the genes of a eukaryotic cell are found inside nucleus. Then Roman 3. State 3 structural differences between DNA and RNA. Very simply, we can say DNA has a double-stranded, DNA is a double-stranded molecule. DNA is a double-stranded molecule. Then on the other side, RNA is a single-stranded molecule. DNA is a double-stranded molecule. RNA is a single-stranded molecule. First difference. Second difference, write both sides. The pathomiliana difference, write both sides correctly. Then second difference, in DNA, pentose sugar is deoxyribose. Here in DNA, you can say pentose sugar is deoxyribose. Pentose sugar is deoxyribose. Deoxyribose. Then on the other side, in RNA, pentose sugar is ribose. In RNA, pentose sugar is ribose. Okay, second difference. Third difference. The method question is it wrong if we write sugar molecule is deoxyribose? It is also correct, but better to say pentose sugar. It is specifically pentose, and pentose sugar is deoxyribose, pentose sugar is ribose. Sugar kiwat kamak nahi, sugar molecule is deoxyribose, sugar molecule is ribose. Okay, that's the second difference. Third difference, DNA has adenine, guanine, cytosine, and thymine nitrogenous bases, and absence of uracil. DNA has adenine, guanine, cytosine, and thymine nitrogenous bases, and absence of uracil. Write the absence of uracil, because recall in the resource book as well as in the note, that is how the differences are given. In DNA, uracil is absent. You have to write that. Then on the other side, you can say RNA has adenine. RNA has adenine, guanine. RNA has adenine, guanine, cytosine, and uracil nitrogenous bases. And absence of thymine. Write the absent nitrogenous base as well. Absence of thymine. That is how differences are given. So use the format as given in your resource book. Okay. Then next, Roman 4. State 3 functions of RNA. State 3 functions of RNA. Then simply you can write the 3 functions of mRNA, tRNA, and rRNA. එහෙම ලියන්න පුළුවන්. You can simply write the functions of mRNA, tRNA, and rRNA here. Okay, so if we will write like that, so as one function we can say mRNA. Then here if you write, when you are writing functions of mRNA, tRNA, and rRNA, together with the function, specifically write which type of RNA. Then don't just say function without mentioning the type of RNA, then it will be incomplete or meaningless. Therefore, write together with the type of RNA. So as a first point, you can say mRNA. mRNA copies genetic information of DNA as a sequence of nitrogenous bases. First point, mRNA copies the genetic information of DNA as a sequence of nitrogenous bases. mRNA copies the genetic information of DNA as a sequence of nitrogenous bases. Also, there's another function of mRNA. You can write that as well. mRNA වලට එකක් ලියන්න පුළුවන්. mRNA, second function of mRNA. mRNA transport genetic information. mRNA transport genetic information from nucleoplasm to ribosome through nucleopore. mRNA transport genetic information from nucleoplasm to ribosome through nucleopore. Transport genetic information from nucleoplasm to ribosome through nucleopore. There, without writing the ribosome, you can say the site of protein synthesis. That way is also correct. Okay, then that's about mRNA. Then tRNA. For tRNA, we can say tRNA transport amino acids to ribosome. tRNA, right, together with the type of RNA there, tRNA transport amino acids to ribosome. Then rRNA, then for rRNA, right, rRNA provides site where polypeptide chains are assembled. rRNA provides site where polypeptide chains are assembled. Provide site where polypeptide chains are assembled. Okay. You can write like that. Then part C1. Name the 3 essential components of a nucleotide. Then it is very easy. Nucleotide has pentose sugar. Then write generally. Pentose sugar or pentose sugar group. Then nitrogenous base. And next, phosphate group. Phosphate group. Pentose sugar group, nitrogenous base, and phosphate group. 3 components of a nucleotide. Okay, next. Ilanka. Name 3 nucleotides other than those found in DNA and RNA and state one function of each of them. So as one nucleotide, we can say, we know there are 4 such nucleotides. One is ATP. ATP function, act as a universal energy carrier. Act as universal energy carrier. Act as a universal energy carrier or act as universal energy carrying molecule. The next, NAD plus. Here you have to write with the plus charge. May writing this plus charge is essential. NAD plus. May plus එක ලියවෙන්න ඕන. May plus එක NAD plus. Otherwise it is wrong. NAD plus functions act as a coenzyme or act as electron carrier. Writing only one is enough here. Act as a coenzyme, act as an electron carrier. And next, act as an oxidizing agent in cellular respiration. Next, act as an oxidizing agent in cellular respiration. Act as an oxidizing agent in cellular respiration. Likewise, next is NADP plus. NADP plus, again, writing this plus charge is essential. Don't just write it as NADP. It should be NADP plus. NADP plus. Again, you can say act as a coenzyme. Act as coenzyme, then act as electron carrier. Act as an electron carrier. Next, act as an oxidizing agent in cellular respiration. Act as an oxidizing agent in cellular respiration. The other one is FAD. NADP plus. NADP plus act as an oxidizing agent in photosynthesis. Here, not cell respiration. NAD plus act as an oxidizing agent in cell respiration. NADP plus, here, this one, NADP plus act as an oxidizing agent in photosynthesis. Act as an oxidizing agent in photosynthesis. Photosynthesis. Next is FAD. Here FAD doesn't contain plus charge. Make a plus charge එකක් නෑ ළමයි. No plus charge. Again, same functions. Act as a coenzyme. Then act as an electron carrier. Also, we can say act as an oxidizing agent in cellular respiration. act as an oxidizing agent in cellular respiration. Hari. Next, name two major substances that are structural components of an eukaryotic chromosome. Those are, one is DNA, other one is proteins. Other structural component, proteins. Generally we can say proteins. In chromatin, there's a specific type of protein called histone. In chromatin it has histone, but generally in chromosomes we can say proteins. Two major substances. Then next, Roman 4. How many chromosomes are found in a nucleus of a human sperm? 23. 23. Half chromosome number compared to a normal cell. 23 chromosomes. 23 chromosomes. හරි. ඊට පස්සේ මෙන්න මේක නම් අපි ඩිස්කස් කරලා කියන්නේ වෙනම ඩෙෆිනිෂන්ස් තියලා නැහැ එහෙම ලොකුවට. මේ මම ඔයගලන් නිකන් කියලා දීලා තියෙනවා මේක ආයි පාරක් ඔය මොලිකියුලර් බයොලොජි වල තියෙන එක. what is a codon කියන එක. මතකද ඔය codons present in mRNA. mRNA එකේ තමයි codons තිබ්බේ. දැන් mRNA molecule එක ගත්තොත් දැන් gene of DNA produces mRNA නේ මතකද? gene of DNA produces mRNA. According to the genetic information, මතක ඇති මෙහෙම gene store genetic information as a specific sequence of nitrogenous bases. According to the nitrogenous base sequence of the gene, mRNA is synthesized. Then during this nitrogenous base sequence of the gene is copied to mRNA as a complementary nucleotide sequence. මතක ඇති නේද? Then mRNA has specific nitrogenous base sequence. mRNA එකේ තියෙන specific nitrogenous base sequence එක. Imagine this is the mRNA molecule. Here along the mRNA it has specific nitrogenous base sequence. මේ වගේ මේ A U G කොහොම තියෙනවා. This is, let's say this is mRNA molecule. Codon එකක් කියලා කිව්වේ මෙහෙම. මේ triplet. This triplet of nitrogenous bases or triplet of nucleotide. Triplet එකක් කියලා කියන්නේ තුනක්. හරිනේ. මේ triplet of nucleotides or triplet of nitrogenous bases is a codon. So the AUG represent one codon. The CCG represent another codon. Then every codon provides signal for specific amino acid. And every codon provides signal for specific amino acid. That means in the ribosome, when protein synthesis occurs, ribosome read this codon sequence. Ribosome read the mRNA codon by codon. When ribosome read one codon, it provides signal for a specific amino acid. Then relevant tRNA molecule carry this amino acid signal by the codon to the ribosome. එහෙම තමයි වෙන්නේ. අපි පස්සේ ආයෙ ඉගෙන ගන්නවා ඒවා. දැනට මෙන්න මෙහෙම ලියමු. ඔතනට කෝඩෝන් කියන්නේ මොකද්ද? කෝඩෝන් is ඔය මොලිකියුලර් බයොලොජි ඉස්සරහට තියෙන්නේ. හරි ලස්සන පාඩමක් මොලිකියුලර් බයොලොජි ඉස්සරහට. ඔය මොලිකියුලර් ලෙවල් එක ඉගෙන ගන්න පුළුවන් ගොඩක් ඒවා මේ සෙල් එක ඇතුලේ. හරි ෂෝක් එක. දැනට මෙහෙම ලියන්න. what is the codon කිව්වම it's a triplet. triplet එකක් කියලා කියනවා. මේ triplet කියන්නේ 3 කියන එක නේ triplet. මේ triplet of nucleotides. Triplet of nucleotides, so you can say triplet of nitrogenous bases. Triplet of nucleotides, or you can say triplet of nitrogenous bases. Triplet of nucleotides, triplet of nitrogenous bases of mRNA. Triplet of nucleotides, triplet of nitrogenous bases of mRNA. As a second point, we can say which provides signal for specific amino acid, or which provides signal for single amino acid කිව්වත් හරි. Which provides signal for specific amino acid, which provides signal for single amino acid. හරි නේද? next ඊට පස්සේ question එක ගන්න Roman 2 වල what is the structural relationship between genes and proteins කියලා. දැන් ඔය බලන්න මෙන්න මේකත් එක්කම ඔක හිතාගත්ත හැකි. දැන් මේ nitrogenous base sequence of mRNA or nucleotide sequence of mRNA is determined according to the nitrogenous base sequence of a gene. අපි මතකද කිව්වා during protein synthesis first step is transcription. During transcription what happens? Nitrogenous base sequence of gene is copied to mRNA as a complementary sequence. ඒතර මෙයාගේ තියෙන්නේ මේ mRNA එකේ තියෙන්නේ gene එකේ තිබ්බ nitrogenous base sequence එකේ complementary copy එකක්. හරිද? එතකොට we can say nitrogenous base sequence of the gene is copied to mRNA as a complementary nitrogenous base sequence. Then this according to this nitrogenous base sequence of mRNA, yeah, amino acids are added to the protein. So ultimately වෙලා තියෙන්නේ මොකද්ද? Ultimately amino acid sequence of the protein. දැන් මෙතන ගත්තොත් එහෙනම් amino acid sequence of the protein is determined by nitrogenous base sequence of mRNA. The nitrogenous base sequence of mRNA depends on the nitrogenous base sequence of the gene. So ultimately, what is the structural relationship between gene and the protein? We can say amino acid sequence of the protein, amino acid sequence of protein is determined by nitrogenous base sequence of gene. Amino acid sequence of protein is determined by amino acid sequence of protein is determined by nitrogenous base sequence of gene. Amino acid sequence of the protein is determined by nitrogenous base sequence of gene. එම කියන්න පුළුවන්. මේක මොකද question එකක් අහනවා. Can we consider the answer as because ribosome is a site of protein since which is made up of genes of DNA. ඔයා කියන මේක මෙහෙම කියනවා because ribosome is a site of protein synthesis which is made up of genes of DNA molecule. නෑ anything. Ribosome එක කොහෙද DNA වලින් හැදිලා තියෙන්නේ? ඕක වැරදිනේ. It is wrong. Ribosome is made up of rRNA and proteins. ඒ ඔලුවට දාගෙන තියෙන information එක වැරදි. Ribosome හැදිලා තියෙන්නේ ribosome each ribosomal subunit is made up of rRNA molecule and proteins. No DNA in ribosome. DNA of nucleolus synthesize rRNA. Then rRNA together with proteins form ribosomal subunits. හරිනේ ඔය ඔලුවට දාන තියෙන එක වැරදි. Ribosome එකේ DNA එකක් ඇති එකක් නැහැ. Ribosome එකේ තියෙන්නේ rRNA එකක්. rRNA plus proteins. හරිද? ඔය දැන් මම කියපු එක තමයි ලියන්න ඕනේ. හරිනේ ඔය මෙතන මේ nitrogenous base sequence of mRNA is determined by nitrogenous base sequence of gene නේ. එතකොට this nitrogenous base sequence of mRNA determines amino acid sequence of protein. එමය වෙන්නේ. එතන මේ codon එකෙන් codon එක 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 amino acid වලට signal දෙනවා. එහෙමනේ යන්නේ. So ultimately what is the structural relationship between gene and the protein? Amino acid sequence of the protein is determined by nitrogenous base sequence of nitrogenous base sequence of gene කියන එක තමයි කියන්නේ. හරිනේ. හරි. ඊගාව 2007. Which one of the following is not a monosaccharide කියලා. It's very easy. Maltose. Maltose is a disaccharide. So answer should be 4. මෙන්න මේක equally easy. All the others are monosaccharides. Then question number 2. Which one of the following substances in plants has mainly a storage function? Starch. Starch has a storage function. Then if you consider the others, glucose is the major respiratory substrate. Cellulose is the main component of plant cell wall. Pectin also component of plant cell wall. Phospholipid is a major type of lipid in cell membrane or plasma membrane. The answer should be starch. Storage function is found in starch. Next, what are biological polymers කියලා. Recall the definition of polymer or macromolecule. කොහොමද කියන්නේ? Molecules with the molecular weight of 10 to the power 4 to 10 to the power 10 grams per mole. බලන්න හරියට. මේක දැන් අපි 100 පාරක් විතර ලියලා ඇති. Biological polymers. What are biological polymers? Polymers, molecules with the molecular weight of 10 to the power 4 to 10 to the power 10 grams per mole. That's the first point. Second point, which are made up of polymerization of a large number of monomers. Which are made up of polymerization of a large number of monomers. Which are made up of polymerization of a large number of monomers. Hari. ඊගාව එක බලන්න, name two major biological polymers found in cells which contain only CHO and indicate one function of each of them. Then we said earlier, among major groups of organic compounds which are act as polymers, one is polysaccharides. I am not telling the answer here directly. මම නිකම්ම explain කරන්නේ බලන්න. polysaccharides ඊට පස්සේ proteins and nucleic acids. These are the polymers. මෙන්න මේවා තමයි nucleic acids. These are the polymers. මේ ටික තමයි polymers උනේ. Among these බලන්න, proteins and nucleic acids contain nitrogen. Then proteins have nitrogen as well as phosphorus. Nucleic acids contain CHO, nitrogen. Proteins have nitrogen and sulfur. Proteins වල තියෙනවා carbon, hydrogen, oxygen, nitrogen and sulfur. Nucleic acids contain carbon, hydrogen, oxygen, nitrogen and phosphorus. Therefore, polymers, biological polymers which contain only CHO should be polysaccharides. Biological polymer that contain only CHO should be polysaccharides. Polysaccharides වෙන්න ඕනේ. Therefore, simply we have to write two polysaccharides here. Biological polymers containing only CHO, polysaccharides. So we can write two polysaccharides. Simply employing starch. Exactly like starch. Function. Simply a storage compound in plants. Storage component or compound in plants. Or there's another way of writing the function of starch. Starch store glucose as an energy source in plants and chlorophytes. Starch store glucose as an energy source in plants and chlorophytes. Another major biological polymer we can say cellulose. Any polysaccharide can be written. Merely a component of plant cell wall. Or else you can write glycogen. Glycogen. Alright, any major polysaccharides as biological polymers containing only CHO. If you write glycogen, then the function is storage compound in animals and fungi. Glycogen act as a storage compound in animals and fungi. Act as a storage compound in animals and fungi. Okay, so write any two polysaccharides. Stored in animals and fungi kiwa 30. Okay, glycogen. Next, Roman 3. Name two major biological polymers. Name two major biological polymers found in cells which contain only CHO, nitrogen and phosphorus and indicate one function of each of them. Carbon, hydrogen, oxygen, nitrogen and phosphorus are found in nucleic acids. Combination nucleic acids. Nucleic acids contain CHO, nitrogen and phosphorus. Nucleic acids. What are the two types of biological polymers? CHO and PTN. One is DNA. We can write two types of nucleic acids. DNA function. There are two functions of DNA. Write one of them. One function of DNA is store and transmit genetic information from one generation to the next generation. One function is store and transmit genetic information from one generation to the next generation. Store and transmit genetic information from one generation to the next generation. Or else you can say, second function of DNA store genetic information for protein synthesis. Second function, DNA store genetic information for protein synthesis. Then second type of biological polymer containing CHONP is RNA. RNA. So as an overall function of RNA, you can say protein synthesis. RNA involving protein synthesis. That way is also correct. If you say involving protein synthesis, that is also correct. RNA involving protein synthesis. Or else you can write a specific function of RNA. We mentioned functions of mRNA, tRNA, rRNA. Or generally we can say synthesis of proteins. It's easy. Next, Roman 4 describe a simple experiment to distinguish sucrose and maltose. Among these, look at this. Sucrose is a non-reducing sugar. Maltose is a reducing sugar. Sucrose is a non-reducing sugar. Maltose is a reducing sugar. Therefore, easily we can distinguish sucrose from maltose using Benedict's test. Benedict's test again lazy and distinguish karagana pulwa. Clear? So write how to carry out Benedict's test. Benedict's test එක ලියන්න තියෙන්නේ. පළවෙනි step එක මොකක්ද කියන්නේ? So we can obtain about 2 milliliters from both solutions into two separate labeled test tubes as A and B. Let's say we obtain 2 milliliters each from the two solutions into two labeled test tubes as A and B. That's the first step. We don't know which one contain maltose, which one contains sucrose. We obtain 2 milliliters each from the solutions given into two separate test tubes labeled as A and B. Here labeling is a must. Otherwise, you will mix up the test tubes. Labeling is a must here. Labeling is a must. Then second step, add equal volume. You know, when we are adding Benedict's, we have to add equal volume. That means when you obtain 2 milliliters from the solution or sample, you have to add 2 milliliters of Benedict's solution. Add 2 milliliters of Benedict's. 2 milliliters of Benedict's. Second step again, add 2 milliliters of Benedict's solution to both test tubes separately. Then we can heat these test tubes in a water bath. Now heat water bath. Heat the test tubes in a water bath. Clear? Gently heat the test tubes in a water bath. Then a solution that produces brick red precipitate. Let's say here brick red precipitate is formed in this test tube. Brick red precipitate. Brick red precipitate. Solution that produces brick red precipitate should be maltose. Maltose, it's a reducing sugar. The solution that doesn't produce a brick red precipitate should be sucrose. Sucrose. Simply we can write like this. Normally, distinguish karagana nathi. Check your points. Points check karagana. Steps points essential. The point obtain 2 milliliters each. Obtain 2 milliliters each from the solutions. Obtain 2 milliliters each from the solutions into two separate test tubes. Into two separate test tubes labeled as A and B. obtain 2 milliliters each from the solutions into two separate test tubes, labeled as A and B. Labeled as A and B. Second step, add equal amounts of Benedict solution. Or you can directly put the value at 2 milliliters of Benedict. Same කියන්න පුළුවන්. Add equal volume of Benedict solution. Remember, definitely we have to add equal volume. Equal volume එකක් Benedict add කරන්න ඕනේ sample එකට. ඒතර ඒකින්ද කියනවා add equal volume of Benedict solution. Add equal volume of Benedict solution. Add equal volume of Benedict solution to the test tubes separately. Add equal volume of Benedict solution to the test tubes separately. Third step, third point. Gently heat the test tubes in a water bath. Gently heat the test tubes in a water bath. Gently heat the test tubes in a water bath. Gently heat the test tubes in a water bath. Next step, ඊළඟ point එක. Here we can write the conclusion together with the observation. The solution that produces a brick red precipitate. Solution that produces or solution that brings brick red precipitate. Solution that brings brick red precipitate is maltose solution. Solution that brings brick red precipitate is maltose solution. Is maltose කියලා ඉවත් ඇති. අනිත් පැත්තට ලියන්නේ the other test tube නැත්නම් the other that doesn't produce a brick red, the other that doesn't produce brick red precipitate is sucrose. The other that doesn't produce brick red precipitate is sucrose. දෙපැත්තම ලියන්න. distinguish කරගන්න කියන්නේ. ඔන්න ඔය ලියන pattern එක තමයි ඔලුවට දාගන්න ඕනේ. labeling කරනවා. ඊට පස්සේ write the procedure correctly. ඊට පස්සේ observation together with the conclusion. ඒවා හරියට තියෙන්න ඕනේ. හරි නේද? හරි ඊගාක බලන්න එනවා 2008 වල අහනවා which of the following cannot be considered as a trace element in plant composition cannot be considered මේක අපිට මේ specifically plant ටික අපි තාම discuss කරලා නැහැ නේ ඉස්සරහට අපි discuss කරනවා ඒතර ඔයාලට තේරෙයි මෙන්න මේ magnesium කියන එක දැනට නම් ඒතර ඔය magnesium අපිට හම්බෙලා නැහැ generally මේක ඉස්සර plant වල නේ එකක් ඉතින් දැන් මේක අමතක කරත් කමක් නැහැ දැනට plant nutrition වල ඉස්සරහට එනවා magnesium. Magnesium is a macro element. Or magnesium is a macronutrient in plant composition. Plant වල magnesium කියන්නේ macronutrient එකක් එහෙම නැත්නම් macro element එකක්. Others are trace. magnesium තමයි එන්නේ. ඒක අපි ඉස්සරහට ඉගෙන ගන්නවා අර generally මේ කතා කරගෙන දැන ටික කොහොමද තියාගෙන. අයිගාවට මෙන්න මේකට යන්න. 2009 බලන්න which of the following substances is found only in animals? Chitin බලන්න chitin is found in fungi. Chitin ඉන්නවා fungi cell wall එකේ. Chitin found in fungi cell wall and arthropod exoskeleton. Since it is found in fungi cell wall, we can't consider. Glycogen also acts as a storage compound in fungi and animals. So it's not the answer. Think where lactose is present. Lactose lactose stored in milk. Milk is produced by mammals. This is found only in animals. Lactose in animals lagi විතරයි. Lactose then according to the syllabus lactose is found only in mammalian milk නේ. එහෙනම් ඒක ගන්න. That is correct. Pectin is found in plants. Middle lamella and plant cell wall contain pectin. So can't consider. Here we haven't discussed hyaluronic acid. මේ hyaluronic acid කියන එක අපි discuss කරලා නැහැ. ඒක අපිට ඉස්සරහදී හම්බෙයි මේ ඉස්සර syllabus එකේ මේක තිබ්බා. monosaccharides polysaccharides වලට. ඉස්සර මේක නිකන් කියනවා. ඒකයි ඔතන කියන්නේ. Hyaluronic acid is actually a polysaccharide derivative. මේක මේ polysaccharide derivative එකක්. It is also found only in animals. මෙයත් ඉන්නේ animals ලගේ විතරයි. Hyaluronic acid is found only in animals. ඒක මම නිකන් කියන්නම් hyaluronic acid කියන්නේ මොකක්ද කියන එක නිකන් ඔයගොල්ලන්ට දැනගන්න විතරක්. Hyaluronic acid is a cementing substance කියලා කියනවා. Hyaluronic acid is a cementing substance. Cementing substance එකක්. It binds cell membranes of adjoining animal cells tightly. ඒ කියන්නේ අපිට මෙහෙම example එක දාමු epithelia වගේ තියෙනකොට නැත්නම් මෙන්න මෙහෙම cells තියෙනවා නම් sorry මේ මෙහෙම හිතමු these are animal cells කියලා. හිතන්න මේ animal cells නා cell membranes of adjoining cells. When I cell membranes, within these cell membranes of adjoining cells are joined together by the cementing substance known as hyaluronic acid. When cell membranes of animal cells are tightly attached together by a cementing substance known as hyaluronic acid. Ek cementing substance එක. Animal cells වල ඔය cell membranes of adjoining cells are tightly attached together by hyaluronic acid. ඒක ඉතින් additionally දැනන් ඉන්න අපිට ඔයවා digest කරන්නේ මේ enzymes එම ඉස්සරහට එනවා. හරි. ඊට පස්සේ structure is a question එක තියෙනවා 2009 වල බලන්න. Part A1. All living organisms show few characteristics which collectively distinguish them from non-living matter. Indicate 4 such characteristics කියලා අහනවා. You can write any 4. There are 7. You can write any 4. Order and organization. Order and organization. තව එකක්. Metabolism. Growth and development. Irritability and coordination. ඊට පස්සේ reproduction. Adaptation. ඔය ඕන එක. And finally heredity and evolution. What do you think? These are the 7 characteristics of living organisms. So write 4. හතරක් ලියන්නයි තියෙන්නේ. Write any 4. Okay, then next. Write down the levels of organization of living matter in correct order. Levels of organization in living matter means hierarchy levels of organization. Levels of organization of living matter means hierarchy levels of organization. In hierarchy levels of organization, there are levels from molecular level up to biosphere level. So smallest level in the hierarchy is molecule level. Molecule. You can use arrows or commas, but write the levels in order. Molecule, then organelle. They should be in order. Cell, then tissue. You can use arrows or commas, it doesn't matter, but they should be in order. Tissue, then organ systems, then organism, then population, community, ecosystem, and biosphere. Normally, clear නේද? all the levels should be written in order. ඔක්කොම order එකට තියෙන්න ඕන. දැන් here මෙතන මේ this is not just system, it should be organ system. මේක organ system කියලා mention වෙන්න ඕන. හරිනේ? ඊගාව බලන්න. Name two polymeric carbohydrates found in living organisms and indicate their monomerous molecules. Two polymeric carbohydrates. It's very easy. You can write any polymeric carbohydrates. Let's say starch. Then monomer is glucose. Also, you can write glycogen. Again, monomer is glucose. ඊට පස්සේ cellulose. Again, monomer glucose. Cellulose is the polymer. Monomer is glucose. Then you can write inulin කියලා ලියුවොත්. Inulin. Monomer is fructose. Monomer is fructose. ඔම ලියන්න පුළුවන්. Okay, write 2. It's easy. Then 2009. බලන්න 2009. Which of the following would be complementary for a DNA strand that contains base sequence of CCA TCG? So what is the complementary base to cytosine? Guanine. Guanine. Then for complementary base to adenine is thymine. For thymine, adenine. The answer is make anything. GGT AGC. GGT AGC. Clear ne? We wrote 2010 essay question. අපි මේක ලිව්වා. We wrote this essay and check in the class. අපි class එකේ මේක check කරගත්තා මුල් කාලෙදිම. ඒක okay එතකොට. In 2011, which of the following is a disaccharide? Is a disaccharide කියලා අහනවා. මොකද්ද මේ තියෙන්නේ? Maltose. Maltose is a disaccharide. Second is the answer. Maltose is a disaccharide. මේක. Which of the following properties of water is directly involved in regulation of body temperature of animals? High specific heat capacity. Then two properties important to moderate temperature. Ability to moderate temperature is given due to two main properties. One is high specific heat capacity. The other one is high heat of vaporization. Among these major property that directly involved in regulation of body temperature is high specific heat capacity. It should be high specific heat capacity. Fourth is the answer. Next. Here this is an ABCD question. Make ABCD question එක. මේකේ ඔයගොල්ලෝ නොදන්න points ටිකකුත් තියෙනවා. බලාගෙන යන්න අපි. Which of the following statement or statements is or are correct on essential elements of plants? They are structural components of plants. Oh, plants වල මේවා structural components කියන එක හරි. Plants are unable to complete their life cycles without these elements. It is also correct. ඒකත් හරි. Then plants show deficiency symptoms when there is low supply of these essential elements. That is also correct. මම කියන්නේ point ඒ වගේ points අපි ඉස්සරහට ඉගෙන ගන්න plant nutrition වලදී. මේකත් එමය. Main source of essential elements is the atmosphere, not atmosphere. Main source of essential elements to plants is soil solution කියලා කියන්නේ. Soil එකෙන් තමයි මේගොල්ලෝ වැඩි හරියක් plants absorb majority of the nutrient elements from soil. Soil solution specifically. ඒක වැරදි. All the essential elements are macronutrients. No, there are trace elements or known as micronutrients as well. So correct statements A, B and C. So according to these directions, we have to use these directions when getting the answer for A, B, C, D questions. A, B and C are correct. According to directions, answer should be 5. You can put 5 here. In the exam, you can mark 5 in the answer sheet. Next. මේක අපි කතා කරා. What are the major characteristics that distinguish living from non-living? In the previous page, we mentioned 7 living characteristics. Again, I am not repeating therefore. ඉස්සෙල්ලා අපි characteristics 7ම කතා කරා. ඒකද මම ආයෙ repeat කරන්නේ. A living matter is composed of living matter is composed of about 92 chemical elements. What are the 6 most abundant chemical elements in living matter? Then if we write four most abundant elements by mass should be oxygen, carbon, hydrogen, and nitrogen. These are the four most abundant elements. Then 6 ගහුවොත් එහෙම මතකද මෙතන කියනවා මේ oxygen, carbon, hydrogen, and nitrogen OCHN account for 96% of living matter. Then most of the remaining 4% is formed by calcium, phosphorus, potassium, and sulfur. හරිද? එතකොට 80 then calcium, phosphorus, potassium, and sulfur are also written according to their mass in living bodies. Therefore, as the 6 most abundant elements, we can write these 6: OCHN, calcium, and phosphorus. OCHN, calcium, and phosphorus. ඔය ටික කොහොම ලියන්න පුළුවන්? හරිනේ මත තියාගන්න ඔය. දැන් මෙන්න මේ පිළිවෙලට යන්නේ. මේ හා තමයි අන්තිකට එන්න ඕනේ. Oxygen, carbon, hydrogen, nitrogen, calcium, phosphorus, potassium, and sulfur. මේ ටික ඔක්කොම පිළිවෙලට තියෙන්නේ. Don't write potassium and sulfur here. මේ දෙක මෙතනට එන්නේ නැහැ. අර ටික විතරයි. Okay, then Roman 2. The most abundant inorganic substance in living matter is water. What are the main functions of water in organisms කියලා අහනවා. දැන් අපි මෙන්න මේ format එකේ discuss කරන්නේ නෑ ළමයි. මේක පටලවා ගන්න එපා මේ බලන්න. We do not discuss in this format. අපි main functions කියලා කියන්නේ නැහැනේ. අපි කොහොමද ඉගෙන ගන්නේ? අපි ඉගෙන ගන්නේ major properties කියලා. We have 4 major properties. Under each property we provide many functions. එහෙමනේ. එහෙම තමයි තියෙන්නේ. ඒතර 4 major functions කියලා අපි ඉගෙන ගන්නේ නැහැ. හරිද? අපි ඉගෙන ගන්නේ 4 major properties කියලා. ඒතර when the question asks to write 4 major functions, don't write 4 major properties. ඒ කියන්නේ ඔයාලා ලියුවොත් එimhe cohesive behavior, ability to moderate temperature, that is wrong. Why? Cohesive behavior, ability to moderate temperature, expansion upon freezing, and versatility as a solvent, those are 4 major properties. A functions නෙමෙයි. හරිද? functions ලියනවා නම් එතකොට අපි තමු under cohesive behavior we are discussing 2 sub properties නේ. One is high addition and cohesion. Due to high addition and cohesion, water perform a function. What is that? Act as a transport medium. ඒවා තමයි functions වෙන්නේ. ඊට පස්සේ due to high surface tension, upper surface water molecules are attracted to lower surface water molecules and water forms a film. අන්න එහෙම. ඊට පස්සේ high specific heat capacity වලට කියනවා water function as a thermal buffer in living systems and aquatic bodies කියලා. ඒවා තමයි ළමයි functions වෙන්න ඕනේ. හරිද? අපි ඒතර අර 4 major functions කියලා එහෙම ඉගෙන ගන්නේ නැහැ. ඒකට ඕන නම් අර function මම කියපු functions වලින් එහෙම 4ක් ලියන්න පුළුවන්. හරිනේ? එහෙම ලියන්න පුළුවන් functions 4ක්. නැතුව මේ 4 major properties ලියන්න එපා. Don't write 4 major properties. හරි ඊගාවට බලන්න එන්න. ඊගාවට කියනවා name 2 major nitrogen containing polymeric compounds. 2 major nitrogen containing polymeric compounds major අහන්නේ in living organisms and indicate 2 major functions of each of them. එකක් ලියන්න පුළුවන් proteins කියලා. Protein contain proteins. Nitrogen containing. අනිත් එක nucleic acids. release in proteins and nucleic acids. Normally, the functions of proteins. There are many functions. You can write any 2. Catalyze biochemical reactions. It was a collagen. Collagen provides support and strength. Keratin prevent desiccation. Hemoglobin transport O2 and CO2. Actin and myosin involved in muscle contraction. Protein functions under any function in the table. Okay. Nucleic acids, right? Function of DNA and RNA. DNA, today we say DNA has 2 functions. DNA store and transmit genetic information from one generation to the next. Store genetic information for protein synthesis. easy. I'm going to make question act as a biological medium for all organisms, a function of water. May act as a biological medium for all organisms. Is it a function of water? No, it's not a function. Water acts as a vital chemical constituent of living cell. Water acts as a vital chemical constituent of living cell, and water provides a biological medium for all organisms. What are those? They are not functions. They are your functions. They are two reasons for the importance of water. Those are the two reasons for the importance of water. They are not functions. Your functions, those are two reasons for the importance of water. Then next, 2012. Which one of the following contains sulfur? Proteins. It's easy. Sulfur is found in proteins. Which one of the following contains sulfur? Proteins. DNA has constituent elements. DNA has carbon, hydrogen, oxygen, nitrogen, and phosphorus. Lipids na CHO. Chitin is a nitrogen containing carbohydrate, CHO nitrogen. Inulin is a polysaccharide, CHO. Sulfur is present in proteins. Proteins contain carbon, hydrogen, oxygen, nitrogen, and sulfur. Protein is the answer. Next. Question number 2. Which of the following regarding carbohydrates is incorrect? Incorrect. Transport within plants, sucrose, correct. Transport within human, glucose. Yes, glucose can be transported in human. Correct. Ape blood wala glucose transport වෙනවා. Storage food in plants, cellulose strong. අන්න වැරදි. Cellulose is a structural component of plant cell wall. Storage food in plants is starch. මේක starch වෙන්න ඕනේ. Component of ATP, ribose, correct. ඒක හරි. මේක හරි. Then component of cell wall of fungi, chitin, correct. So answer should be 3. Third is the answer. Again, there's an ABCD question. මේකත් දැන් ABCD question එකක් බලන්න. Which of the following carbohydrate or carbohydrates give a positive reaction in Benedict test කියලා අහනවා. මතක් කරන්න, reducing sugars. Reducing sugars produces a positive reaction. මොකද්ද positive reaction එක? Reducing sugars produce brick red precipitate. That is a positive reaction. So what are the reducing sugars? All monosaccharides are reducing sugars. All monosaccharides are reducing sugars. Then among disaccharides, maltose and lactose are reducing sugars. All monosaccharides, then maltose and lactose. So lactose, then glucose, maltose, ribose. Ribose is also monosaccharide, reducing sugar. A, B, D, and E. A, B, D, E means answer should be 5. You can put number 5 here. Positive reaction, brick red precipitate is given by all monosaccharides and maltose and lactose. Within A, B, D, E. Answer 5. මෙන්න මේ question එක ඔයගොල්ලෝ ලියන්න ගෙදරදී මේ මම එක ක්ලාස් එකකදී නම් කිව්වා මෙන්න මේ question එක ලියන්න කියලා. Describe the basic chemical nature and functions of protein. 2013 question එක නිකම් වලට කිව්වා මේක ලියන්න කියලා. අනිත් ලියන්න මේක. Describe the basic chemical nature and functions of proteins. ඒක ලියන්න. ඒක අපි ක්ලාස් එකේදී බලමු මේ marking scheme එකකට මේක ලියලා තියන්න. Then 2014, which one of the following represents the composition of elements of albumin? Albumin is a protein. Albumin can a protein. Albumin is a protein. So what are the components of albumin? Methane 3. Protein එකක්නේ, carbon, hydrogen, oxygen, nitrogen, and sulfur. Then next, which one of the following chemical tests can be used to show the presence of glucose in a solution? Glucose is a reducing sugar. Reducing sugar එකක්නේ. It can be identified using Benedict's test. Glucose is a reducing sugar. Then we can do Benedict test. Benedict test. You are lazy. Next. බලන්න part A1. What is an water is an essential ingredient for life, and it is the most abundant inorganic compound in living matter. That is true. State two major functions of water in living organisms. Again, they ask to write two major functions. Again, don't write major properties. I paragraph properties ලියන්න එපා. Don't write properties. Major functions. But on a functions ලියත හැකි act as a transport medium, act as a thermal buffer. ඒ වගේව ලියන්න පුළුවන් ඕන. Act as a transport medium, act as a thermal buffer in living systems and aquatic bodies. Those are okay. ඒ වගේව ලියත හැකි. Don't write properties. මේක ලියත හැකි වෙන්නේ Roman 2. Name two properties of water. Name two properties of water which help to moderate changes in temperature in living organisms. මොකක්ද එකක්? High specific heat capacity. High specific heat capacity. Next, high heat of vaporization. High heat of vaporization. One is high specific heat capacity. Next, high heat of vaporization. Write with the word high specific heat capacity and high heat of vaporization. You cannot name two disaccharides which are reducing sugars. Maltose and lactose. Maltose and lactose. Those are the two reducing sugars, maltose and lactose. The most enzymes become, under Roman 4, most enzymes become biologically inactive at high temperatures due to denaturation. State two physical changes of the enzyme molecule that contribute to this කියලා කියන. දැන් මෙන්න මේක අපිට ඇතර මෙතනදී ලොකුට කතා කරන්න බෑ මේ enzymes පාඩමේ කතා කරන්නේ. ඔය අපි කියනවා මේ during denaturation shape of the three-dimensional shape of the enzyme molecule changes කියලා. ඒතර ඒක physically observe කරන්න පුළුවන් change එකක්. So we can write it as one physical change. මොකක්ද එකක් ලියනවා එතකොට alteration නැත්නම් alteration of shape of the enzyme molecule. During denaturation, shape of the enzyme molecule changes. During denaturation, then enzymes are proteins. Enzymes are proteins. Then during denaturation, weak chemical bonds and interactions will be altered. When weak chemical bonds and interactions alter, shape of the protein or three-dimensional shape of protein will be lost. Alteration of shape කියන්න පුළුවන් නැත්නම් loss of specific three-dimensional shape. Loss of specific three-dimensional shape. One physically observable change. ලියක බලන්න පුළුවන්. ඊට පස්සේ අපි පස්සේ ඉගෙන ගන්න මේක දැන් ඔයාලට ලියත හැකි කෙලින්ම මේක ඩිරෙක්ට්ලි ලියන්න පුළුවන්. Then another one මේක තමයි ඔයාලට ලියන්න බැරි එක දැනට. ඔය enzymes වල තියෙනවා enzyme has a specific site called active site. So active site is a place where catalytic reaction takes place. එතන තමයි reaction එක වෙන තැන. ඔන්න active site එකට තියෙනවා specific shape එකක්. So when the shape of the enzyme molecule changes, shape of the active site also changes. So the second එක physically observe කරන්න පුළුවන් change එක විදිහට ලියනවා changes the shape of active site කියලා. Changes the shape of active site. And it was Roman 4. Name the three components of a nucleotide. මේක දැන් 100 වතාවක් විතර කිව්වා අපි. මොනවද? pentose sugar, nitrogenous base and phosphate group. Pentose sugar, nitrogenous base and phosphate group. ඒක ලේසිනේ. That's 2015. බලන්න which of the following chemical element is the most abundant in living organisms by mass? Oxygen. mass එකෙන් වැඩිම තියෙන oxygen. අපි දැන් order එක ලියුවේ ඉස්සෙල්ලා. most abundant elements in order by mass: oxygen, carbon, hydrogen and nitrogen. කොහොමනේ පිළිවෙල එන්නේ? Oxygen is the most abundant one. Very easy. Then problem 2, sorry, second question. Which of the following polymers is found only in plants? Plants වල විතරක් තියෙන කවුද? පටගාලා කීපයක් දෙන්න. මේ inulin, inulin. Inulin කොහෙද තියෙන්නේ? Inulin store in dahlia tubers. Inulin store in dahlia tubers. Inulin should be the answer. So glycogen ගත්තොත් glycogen found in animals and fungi. මේක fungi වලත් තියෙනවා. This is found in fungi as well as in animals. Chitin කිව්වොත් chitin found in fungal cell wall and arthropod exoskeleton. RNA, RNA ribonucleic acid ne RNA. It is found in any organism. Keratin animals lage තියෙනවා. Keratin found in animals. The answer is 4. And මේක බලන්න මේක direct question එකක් නෙමෙයි 41 ABCD question එකක්. We can use those directions to get the answer. Get the answer. බලන්න මේක this is not a direct theory. මේක direct theory එකක් නෙමෙයි. මේක බලන්න අහන එක carbohydrates are commonly stored as starch in plant storage organs කියලා කියනවා. ඒ fact එකක්. ඒක දාන්න එකක්. ඊට පස්සේ අහනවා which of the following properties of starch makes it a useful storage material. ඒ කියන්නේ that means which of these properties helpful starch to act as a storage material කියලායි අහන්නේ. ඒතර storage material එකක් විතර වැඩ කරන්න නම් මොන වගේ properties තියෙන්න ඕනේ? මොන වගේ properties ද? දැන් එක අපි හිතමු ඔන්න cell එකක් ඇතුලේ within a cell it has aqueous medium නේ cell එක ඇතුලේ තියෙන aqueous medium එක. If starch is soluble then it can't be easily stored. So exceptions තියෙනවා. දැන් inulin is a storage polysaccharide. It is an exception. Even though it's a storage polysaccharide, it can store. It's an exception. But normally most of the storage compounds are insoluble in water. So water insolubility is one good feature that present in starch. There are water insoluble වුණාම कारण it is water insoluble, it can easily store in an aqueous medium. Aqueous medium එක easily store කරන්න පුළුවන්. ඒක dissolve වෙන්නේ නැහැ. ඒක හොඳ property එකක් වෙනවා. අනිත් එක තමයි හිතන්න if a storage compound is highly active, chemically active නම් metabolically active නම් එතකොට මොකද වෙන්නේ? Then it will directly react with some other substances. එයා ගොඩක් reactive එක්කෙනෙක් නම් ටක් ගාලා අනිත් අයත් එක්ක react කරනවා. එතකොට එයාව store කරලා තියාගන්න බෑ. store කරලා තියෙනකොට එයා වටේ පිටේ ඉන්න අයත් එක්ක react කරනවා. ඒකද chemically non reactive කියන එකත් ගොඩක් important property එකක් වෙනවා. හරිද? එතකොට මෙන්න මේකේ බලන්න මෙන්න මේ C and D. C කියලා කියනවා it is chemically non reactive. It is an important property to act as a good storage material. And it is insoluble in water. It is also very important. ඒකත් important. So C and D ගන්න පුළුවන්. C and D important properties to act as a storage material. Osmotically inactive. Osmotically inactive කියලා කියන්නේ osmosis වලට involve වෙන්නේ නැහැ කියන එක නැත්නම් dissolve වෙන්නේ නැතිඳ osmosis වලට effect එකක් නැහැ කියන එක. ඒකේ fact ඒක storage material එකක් විදියට තියෙන්න එහෙම significance එකක් importance එකක් නැහැ ඒකේ. Insoluble in water කිව්වහම හරි. Translocate ඊට පස්සේ second B වල කියන්නේ it is easily translocated කියලා. ඒක වැරදිනේ කොහොමද? Starch is not translocated. Starch නෙමෙයි translocate කරන්නේ. Sucrose is a translocating carbohydrate. Sucrose තමයි translocate කරන්නේ. ඒතර කොහොමත් ඒක වැරදි. ඒතර අනිත් එක E වල කියනවා macromolecule. Being a macromolecule is not important to act as a storage compound. Macromolecule එකක් වෙන එක important නැහැ storage compound එකක් විතර වැඩ කරන්න. මොකද අපි දැන් sucrose ගත්තා. Sucrose is also storage compound. ඒ කියන්නේ being a macromolecule is not important to act as a storage compound. So answer should be C and D. C and D means according to directions in ABCD questions we can put number 4 as the answer. Number 4 තමයි ලියන්නේ answer එක විදියට. හරිද? ඊගාව එක structure ඉස්සරහට what is the most abundant group of biological molecule? Carbohydrates. Carbohydrates. Group of biological molecule, carbohydrates. They name the nitrogen containing structural polymer present in exoskeleton of some animals, chitin. So name an oxidative disaccharide. මේ බලන්න මේ අහන විදිය. පාසිෆ් එකේ name an oxidative disaccharide කියලා අහනවා. ඒ කියන්නේ දැන් ඔන්න මම ඔයගලන්ට කියලා දුන්නේ නේද මතකද? මේ ඒ කියන්නේ අර තේරුම් අරන් ඉන්න එක වැදගත් කියන්නේ ඒකයි. දැන් අපි කියනවානේ reducing sugars කියලා. හරිද? reducing sugar කියලා කියන්නේ එතකොට reducing sugar එකක් නම් එයා reducing sugar එකක් විදිහට මොකක්ද යාගේ තියෙන property එක? Reducing sugar means it can reduce another substance by oxidizing itself නේ. තමන් oxidize වෙලා අනිත් එක්කෙනෙක් reduce කරන්න පුළුවන් කෙනා reducing agent එකක්, reducing sugar එකක්. ඒතර මේ බලන්න මේක යහන්නේ ඒ වගේ එකක් නේ. name an oxidative disaccharide. ඒ කියන්නේ disaccharide which can oxidize itself but reduce another substance. Oxidative disaccharide එකක්. හරිද? Oxidative disaccharide එකක් නේ. ඒතර ඒකට reducing sugar එකක්ම තමයි අහන්නේ. ඒතර reducing disaccharide එකක්. reducing sugar එකක් විදිහට අඩ කරන disaccharide එක. oxidative disaccharide එකක් කියලා කියන්නේ එයාට පුළුවන් වෙන්න ඕනේ. හරි නේද? තමන් oxidize වෙලා අනිත් එක්කෙනාව reduce කරන්න. oxidative. ඒතර අපිට ලියත හැකි maltose, lactose. maltose ලියන්න පුළුවන්. lactose can be written. ඒ oxidative disaccharides. ඊට මේ අනිත් පැත්තට non reducing disaccharide sucrose. Non-reducing disaccharide sucrose. That means it can't oxidize. Sucrose can't oxidize. Sucrose will oxidize in the bed. So it is not a reducing sugar. Reducing sugar can reduce another substance by oxidizing itself. So they are oxidative. They are oxidized. Non-reducing sucrose. Iris Roman 4 may using a suitable diagram show how a peptide bond is formed in between two amino acid molecules. Peptide bond එකක් කොහොමද හැදෙන්නේ කියලා අහනවා. ඕක මේ අර අපි weekly test paper එකකදීත් ඇඳලා පින්නවා. මම ඕක තමයි කිව්වේ past papers වලදී දීලා තිබ්බයි කියලා ඔය question එක. past papers වලදී දීලා තිබ්බා peptide bond මේ peptide bond is formed dipeptide එකක් හැදෙන හැටි අහන්නේ. Using a suitable diagram show how a peptide bond is formed between two amino acids molecules කියලා නහන්නේ. ඒක easily මෙහෙම අඳින්න පුළුවන්. Draw two amino acids correctly and show how condensation reaction occurs to form the peptide bond. වෙනන කොහොමද condensation reaction එක වෙන්නේ peptide bond එක හැදෙන්න. මෙහෙම අඳින්න මේ. This is one amino acid. Carbon atom to which carboxyl group, hydrogen atom, variable alkyl group, and amino group is attached. This is one amino acid. Then draw an adjoining amino acid. This is an adjoining amino acid. Alkyl group, carboxyl group, hydrogen atom, and amino group attached here. Then when forming the peptide bond, condensation reaction occurs. Condensation reaction occurs between this OH group of the carboxyl group of one amino acid together with this hydrogen atom of the amino group of adjoining amino acid. This is removed as water. Show the removal of water molecule by condensation reaction like this. Then draw the formation of peptide bond. It has a peptide bond එක ඇඳලා පෙන්නන්න. Here now I am drawing the remaining parts of the first amino acid. So පළවෙනි එකේ යන්දිනවා මෙහෙම. Then here this is the peptide bond. මෙතන හැදෙන peptide bond එක. This is a peptide bond. It is formed between the carbon atom of carboxyl group of one amino acid and the nitrogen atom of the amino group of adjoining amino acid. Then show the removal of water molecule as a product. So we can label this as the peptide bond. Meaning this is the peptide bond. ඔන්නෝම draw කරලා පෙන්නන්න පුළුවන්. Draw this and show how peptide bond is formed. Clear නේද? හරිනේ? එන ඊළඟ එක. Next. බලන්න එහෙනම් what is the test used to determine the presence of peptide bond in proteins? බලන්න අපි ඔය කියලා දීලා තියෙනවා. What is that? Biuret test. ඒකට කියන්නේ biuret test. මතකනේ? අර alkaline copper sulfate. ඒ කියන්නේ potassium hydroxide with copper sulfate is added. That is biuret. Biuret test. මතකද මම කියලා දුන්නා? Biuret is a specific test to identify the presence of peptide bond. Therefore, we said that not only a protein, any compound containing peptide bond will produce a purple color with biuret. Biuret means alkaline copper sulfate. ඒ කියන්නේ potassium hydroxide with copper sulfate. Then Roman 5a. It says what is a glycosidic bond? Recall the common pattern that we use in defining a bond. So we can say that is a bond. Glycosidic bond is a bond that joins two monosaccharide molecules together. A bond that joins two monosaccharide molecules together. That's the first point. A bond that joins two monosaccharide molecules together. A bond that joins two monosaccharide molecules together. Second point, by condensation reaction. Second point, by condensation reaction, removing a water molecule. By condensation reaction, removing a water molecule. There in the second point, you have to mention both condensation reaction and removal of water molecule. සම්පූර්ණයෙන් කියන්න ඕනේ. Next, simple B of the same question. Name two biological compounds containing glycosidic bond. You can write any disaccharide or polysaccharides like disaccharide ලියන්න පුළුවන් ඔයාට ඕන එකක්. Maltose, lactose, sucrose. Any disaccharide or write a polysaccharide like starch, glycogen, cellulose, inulin. All of them have glycosidic bonds. ඕන එකක්. I think very easy. හරි ඊට පස්සේ what are the three chemical constituents of a nucleotide? We repeated this several times. Pentose sugar, nitrogenous base and phosphate group. Pentose sugar, nitrogenous base and phosphate group. Question එකක් අහන්න. මේක කරනවා is it wrong if we write disaccharide and monosaccharide? Monosaccharide කිව්වොත් නම් කොහොමත් වැරදි. දැන් මේක යහන්නේ name two biological compounds නේ. Compound name කරන්න කිව්වම write name them. එතකොට කොහොමත් monosaccharide කියන එකේ glycosidic bonds නැහැ. Monosaccharide do not have glycosidic bonds. It is completely wrong. Monosaccharides को हमें glycosidic bonds नहीं, it is completely wrong. Two biological compounds को हम you can name two example biological compounds. Any disaccharide or any polysaccharide can be named here. Any disaccharide or major polysaccharide. මොනොසැකරයිඩ් කොහොමත් වැරදි. මොනොසැකරයිඩ් වල glycosidic bonds නැහැ. ඩයිසැකරයිඩ් හෝ පොලිසැකරයිඩ් වෙන්න ඕනේ. හරි ඊට පස්සේ what are the three chemical constituents of a nucleotide? ඒක අපි ඉස්සෙල්ලා කිව්වා pentose sugar, nitrogenous base and phosphate group. ඒක ලේසි. ඊට පස්සේ Roman 7 name three nucleotides and state one function of each of them. ඒකත් අපි අර ගොඩක් කිව්වා. You can write nucleotides that do not form nucleic acids. ATP, NAD+, NADP+. We mentioned those today. අපි ඒකින්ද එක skip කරමු. ඒ ටික අපි අද කතා කරා. මේකත් ඕකේ මේ what are macromolecules? මොකද පළවෙනි point එක? Molecules with the molecular weight of 10 to the power 4 to 10 to the power 10 grams per mole, which are made up of polymerization of a large number of monomers. ඒවා අපි repeatedly ආපුවා අපි ඉස්සෙල්ලා කතා කරා ඒවා. ඊගෙන මම ලොගෝට ආයවා කියන්නේ නැහැ. අදම කතා කරා ඒවා. And then Roman 2 name three types of macromolecules found in living organisms. ඒක ලියන්න පුළුවන්. What are the three types of macromolecules? ඉස්සෙල්ලා අපි කිව්වා one is polysaccharide. We do not write it as carbohydrates because among carbohydrates, monosaccharides and disaccharides are not macromolecules. So polysaccharide, proteins, and nucleic acids. Now make balance. Name the disaccharide sugar found in the following and indicate the constituent monosaccharide unit of each of them. Sugarcane plant nang, what's the disaccharide present? Sucrose. Sucrose is the disaccharide present. Monosaccharide subunits are, right, both glucose and fructose. They both should be written. Monosaccharide subunits, glucose and fructose. Germinating seeds contain maltose. Maltose will be present during seed germination. Amylase enzyme hydrolyze starch into maltose. Amylase enzyme hydrolyze starch into maltose. The germinating seeds have maltose. Sugars present glucose. Two glucose molecules together form maltose. Monosaccharide subunit glucose. Then milk contain lactose. Milk wala තියෙනවා lactose. Then monosaccharide units glucose and galactose. Right, both glucose and galactose. Then next, what is the monosaccharide unit found in NAD and ATP? That is ribose. Ribose pentose sugar. Both NAD and ATP contain ribose. Can we write ribonucleotide and deoxyribonucleotide as examples for nucleotides? එහෙම ලියන්න පුළුවන් ඉතින්. එහෙම ලියුවොත් එහෙම ribonucleotide කිලිවොත් if you write ribonucleotide for the previous one මේ අහන්නේ ඉස්සෙල් එකේ two nucleotides ලියන්න කිව්වේ ribonucleotides කිව්වොත් function එක ලියන්න monomer of RNA. Deoxyribonucleotide monomer of DNA. That is also correct. හරි ඊට පස්සේ මෙන්න මෙතන question එකට එක්කෙනෙක් අහලා තිබ්බා write the monosaccharide unit found in NAD and ATP pentose කියලා ලියන්න පුළුවන්ද කියලා. monosaccharide unit එක specifically කියන්න පුළුවන් නේ අපිට. pentose කිව්වම common වෙනවා. pentoses there are three types of pentoses ribose, deoxyribose, ribulose කියලා commonly තුනක් තියෙනවා. දැන් NAD plus ATP මේ මොනවද මේ these are the nucleotides that do not form nucleic acids. These are the nucleotides that do not form nucleic acids. Nucleic acids හදන්න නැති ඔය කියන්නේ. එතකොට මතකද මම කිව්වා ඔයගලන්ට in nucleotides that do not form nucleic acids pentose is ribose. Pentose එක ribose. So specifically write the monosaccharide unit here as ribose. Ribose pentose sugar. එහෙම ලියවන වඩා හොඳයි. Or at least write ribose. හරිද? ඊගාව ඒ කියනවා lipids are one of the major organic compounds in living organisms State two important characteristics of lipids which distinguish them from other major biological molecules. Other major biological molecules are carbohydrates, proteins, and nucleic acids. So how could we distinguish lipids from carbohydrates, proteins, and nucleic acids? One major feature, one distinguishable feature in lipids is they are hydrophobic. Hydrophobic molecules. It is one of the major features. Hydrophobic molecules. Lipids are hydrophobic. Diverse group of hydrophobic molecules. Then ester bonds. The ester bonds are not common to every type of lipid. The steroids do not have ester bonds. Hydrogen is to oxygen ratio is not 2:1. Hydrogen is to oxygen ratio is not 2:1. Hydrogen is to oxygen ratio is not 2:1. Or you can say lipids have higher content of hydrogen and comparatively lesser content of oxygen. That is also correct. Hydrogen is to oxygen ratio is not 2:1. Or else you can say lipids have higher content of hydrogen and lesser content of oxygen. And those are common to every type of lipid, and using this we can distinguish lipids. ඒ දෙක වඩා හොඳයි. මේක distinguish කරන්න පුළුවන්. ඊට පස්සේ මෙන්න මේ ඊළඟ කියන Roman 6 වල name 5 major types of lipids කියලා. In our syllabus we do not discuss 5 major types. දැන් අපි දැන් syllabus එකේ 5 major types discuss කරන්නේ නැහැ නේ. පරණ එකේ නම් එහෙම කිව්වා. අපි 5 discuss කරන්නේ අපි ඉගෙන ගන්නේ 3 major types of lipids කියලා. අපි මේක ඉගෙන ගන්නවා 3 major types. Major types තුනක් තමයි ඉගෙන ගන්නේ. මොනවද එතකොට? අපි ඒ තුන ලියත හැකි. Fats, phospholipids and steroids කියලා ඉගෙන ගන්න. Fats, phospholipids and steroids. ඔයිට අමතරව ඉස්සර syllabus එකේ ඉගෙනුවා terpenes, wax වගේ ඒවා. ඒවා තමයි ඊට අමතරව major types of lipids කියලා කිව්වේ. ඒක මේ දැන් ලියන්න ඕනේ. ඔය major types of lipids කියලා කියන්නේ most abundant type of lipid. ඒ කියන්නේ major කිව්වම වැඩිපුර තියෙන ඒවා. Major types of lipids වැඩිපුර තියෙන ඒවා. වැඩියෙන් තියෙන ඒවා. එතකොට ඔය තුන තමයි most abundant type of lipids. 3. තුනයි ලියන්න වෙන්නේ. ආලම මෙන්න මේකත් ලියන්න මේ essay එකක් මේකත් protein එකක්. මේකත් මම ලියන්න කියලා කිව්වා මේ එක class එක. අනිත් අයත් මේක ලියන්න. ගේ සතිය කිව්වේ describe the basic chemical nature and general structure of proteins. ඒ ඉස්සෙල්ලක තිබ්බේ chemical nature and functions. මේක general structure, chemical nature and general structure. ඒ දෙක ලියන්න. අපි proteins වල essays මොකද වැඩි ලියුවේ නැහැ, මොකුත් ලියුවේ නැහැ. ඒකින්ද ඒ දෙක ලියන්න. හරි නේද? හරි එහෙනම් අපි අද රොතේ නවත්තමු. ඊටු තව 2016 ඉඳන් 24 වෙනකම් papers 8ක් තියෙනවා. අපි මෙතනින් අද නවත්තමු. එතන 11යි නේ ඔයගලන්ට කොහොමත්. එතකොට අමාරුයි දැන් මේ ආපු අය දැන් ටික ටික යනවා. මේ නිදිමත හැදෙයි. ඒකින්ද අපි ඊළඟ දවසේ මේක ඉතුරු ටික ඩිස්කස් කරමු. ඉතුරු papers 8 තව දවසක ඊළඟ බ්‍රහස්පතින්ද වගේ ඊළඟට ඩිස්කස් කරමු. පොඩ එතකොට හැමදාම පොඩ මතක ඉන්නවා. හරි. එහෙම කරමු. හැමදාම ටික ටික ටච් වෙනවා. පාඩම් කරගන්නවා cellular basis of life වල අපි ඩිස්කස් කරන් යන ටික හොඳට පාඩම් කරගන්න. එතකොට මේ ඒකෙත් අපිට එතකොට cellular basis of life, cell division දෙකම ඩිස්කස් කරලා පස්සේ මේ විදිහට දිගටම ඒකෙත් past papers ටික ඩිස්කස් කරගෙන යන්න පුළුවන්. මේ කොටසෙන් කොටස. අතට හැමදෙස්සෙම ටච් එක ඊට හැකි. හරි. එහෙනම් good night. ඊළඟ දවසේ හම්බෙමු.